Please mute your phones. Please mute your voice. Please mute your videos. We are going to start in about 1 or 2 minutes. If you have any questions in the middle of the session, please type it in the chat box. So we are going to address those. We are going to interact with the guest speaker at the end of the session. If you have any questions or any doubts, please message in the chat box. We are going to interact with the guest speaker at the end of the session.
YouTube is live, sir. You can start the session.
So welcome you all for this 6th day of the Zoonoses Day webinar series, and also to the celebration of Zoonoses Day. Before we start the webinar, it's my turn to formally welcome you all. First of all, it's my privilege to welcome our Honorable Vice Chancellor, Professor H.D. Narayan, the invited guest of this occasion. On behalf of all the participants and on behalf of all the members of the organizing team, from the bottom of my heart, I welcome Professor Honorable Vice Chancellor, Professor H.D. Narayan, sir. I wholeheartedly welcome our respected Dean, Professor M.C. Shivkumar, sir, and also the Chairman of the Organizing Committee, and all the officers of the Karnataka Veterinary Animal and Fishery Sciences University for the celebration of this Zoonoses Day. I cordially welcome our invited speaker, Dr. Jagadish Hiremath, Senior Scientist and Biosafety Officer, Nivedi, Bengaluru, to this Zoonoses Day celebration, and also to present his talk. Welcome, sir. Thank you. I cordially welcome all the faculty members of Karnataka Veterinary Animal and Fishery Sciences University, and all those teachers, fellow professionals, and colleagues who are participating in this webinar series all across the country. I welcome you all. I take this opportunity to welcome all the scientists, the veterinary doctors, medical doctors, and students across the country to this webinar series. I welcome you all once again. I take this opportunity to read out the message that was sent by our Honorable Vice Chancellor. Sir is busy, so due to unavoidable circumstances, he could not enter into the webinar. However, he has sent a message, so I take this opportunity to read it. Dear Deans, Directors, Officers, and colleagues of the University, invited speakers, speaker of today's Zoonoses lecture, faculty members of various universities across the country, scientists, professionals, dear students, and friends. As you are aware, World Zoonoses Day is celebrated every year to mark the historic event of preventive healthcare, prevention of infectious diseases. This day, 135 years ago, legendary microbiologist and French biologist Louis successfully vaccinated Joseph Mister against rabies and saved him from this deadly disease. Friends, there are 1,415 infections of humans that are known to the mankind, of which 838 are having origin in animals. These dreaded diseases, as rightly termed as zoonoses by the legendary cellular pathologist, Sir Rudolf Virchow, continue to affect the lives and well-being of humans and varied animal species since time immemorial. It is of immense public health concern that about 75% of recently emerging infections are zoonotic, and three-fourths of such infections have wildlife origin. Changing population dynamics, climate change, vector biology, above all, change in the human lifestyles have all significantly contributed to the burden of ever increasing zoonoses. Friends, during this time of COVID 19, wherein the world is severely affected by the pandemic, we all must learn the lesson to work under one health umbrella and strengthen interdisciplinary and intersectoral collaboration to combat such zoonoses in the future. It is timely that our university is proactive in addressing some of these major zoonotic diseases, such as rabies, foodborne infections, anthrax, paratuberculosis, Casimir forest disease, etc. I personally appreciate the efforts initiated by Dr. MC Shivakumar, Dean Veterinary College, Hassan, and his team for organizing this national webinar series and Zoonosis Week. I hope the participants will definitely be benefited by these deliberations held during this week-long webinar series. Thank you. Jai Hind. So thank you very much, sir. That was the message that was sent by our Honorable Vice Chancellor, Professor H.D. Narayan-Swamy, sir. Thank you very much, sir, for your guidance and direction in organizing this national webinar series. Thank you very much, sir. So now we'll move on to the actual the presentation of the today. Before that, I would like to briefly introduce the invited speaker, the guest speaker, Dr. Jagadish Hiremath, Senior Scientist and Biosafety Officer, ICAR National Institute of Veterinary Epidemiology and Disease Informatics, Bengaluru. Research interests of Dr. Jagadish include immunoepidemiology of porcine, respiratory viral diseases, vaccine efficacy, vaccine effectiveness, vaccine failures, laboratory biosafety and biosecurity, and biorisk assessment. His education he completed his BVSc and AH from Veterinary College, BIDAR, US, Harvard in the year 2003. And he completed his MBSc from GB Pant University of Agriculture and Technology, Pantnagar, Uttaranchal in 2005. And he completed his PhD from College of Veterinary Medicine, the Ohio State University, Columbus, USA. And various positions held by Dr. Jagadish Hiremath include, he worked as a research fellow from March 2006 to March 2007 at CSIR, Center for Cellular and Molecular Biology, Hyderabad. And he worked as assistant professor in the Department of Veterinary Biochemistry between March 2007 and April 2009 in Veterinary College, Shivamogga, Karnataka Veterinary Animal and Fishery Sciences University, Bidar, Karnataka. From April 2009 till date, Dr. Jagadish Hiremath is working as scientist and later as a senior scientist in ICAR National Institute of Veterinary Epidemiology and Disease Informatics, Yelahanka, Bengaluru. His expertise in the area of laboratory biosafety and biosecurity include, he worked as a biosafety officer, has been working as biosafety officer in ICAR Nivedi. He is instrumental in developing biosafety manual, safety practices, and implementation of national safety guidelines. He is also in charge of Biosafety Laboratory 2++ ICAR Nivedi. He is also working as DBT nominee for IBSC in Pentavalent Bioscience Private Limited. He is also a member secretary of IBSC and ICAR Nivedi. He has developed training modules in laboratory biosafety and biosecurity from 1 day to 5 days duration and national and international capacity building training programs in the area of laboratory biosafety and biosecurity. He has developed Indian biosafety application, a mobile application to assist in agent classification and risk group categorization. The honors and awards received by Dr. Jagadish Hiremath, the American Association of Veterinary Immunologists, American Association of Immunologists has conferred him with the Young Investigator Award at the Conference of Research Workers in Animal Diseases in 2013. OARDC Professional Growth Scholarship from Ohio Agricultural Research and Development Center, Worcester, USA. He is also recipient of the ICAR International Fellowship between 2010 and 11 to pursue PhD at the Ohio State University, USA. He secured second rank in agricultural research services of the NET examination held in the year 2008-9. Dr. Jagadish Hiremath has published more than 15 international publications, full-length papers, and also 5 national research papers. On behalf of all the participants, I welcome once again to Dr. Jagadish Hiremath on behalf of Karnataka Veterinary Animal and Fishery Sciences University. And without much delay, now I hand over this webinar platform to Dr. Jagadish to present his talk on epidemiology of emerging and animal coronaviruses. Over to Dr. Jagadish.
Thank you, Dr. Girish, for the nice introduction, and my sincere thanks to Honorable VC KVAFSU, Dr. H.D. Narayanaswamy sir, and my sincere thanks to Dean Dr. M.C. Shivakumar sir, and I also thank all my senior teachers, colleagues, and fellow friends who are working in that university. I have been the student of that university, and I also worked as a faculty for a period of 2 years in that university. And coming to the topic as such, hopefully I'm audible.
Yes, yes, you can please continue.
Okay, so now. So, on the eve of World Zoonoses Day, today we have a topic on epidemiology of emerging coronaviruses in human and animals. As you all know that coronaviruses are been very known and notorious in a sense. They have been responsible for emergence of some of the biggest and dangerous viruses recently, and currently as we are undergoing the pandemic of coronavirus, this topic becomes more relevant in order to understand what these viruses are, why these viruses every time cause so much of damage, what they are causing, what is the reason why they are able to do it, and what is its impact on human, and what is its impact. Use mask, try to find a place which is safe to do exercise without mask. So upfront, coming to the epidemiology of COVID-19 as such. So when you talk about epidemiology, so the epidemiology triangle is one thing which comes to our mind, which we have been exposed to right from our BVSC. And there are 3 integral factors which will play a very, very important role, and they come together to cause an outbreak of a disease. Environment is very, very important. Host susceptibility and host factors are very, very important. And comes the agent. So how is the agent? What are its features? What is its virulence? What is its pathogenicity? So those are all the factors of agent which will decide whether a disease is going to be spreading in the form of endemic, pandemic, epidemic. So there are various ways of explaining a disease outbreak, and depend upon these combination of factors, it will decide what is the extent of disease. So just to define, we all been hearing what is like emerging diseases, re-emerging diseases, exotic diseases, transboundary animal diseases. So these are all terminologies which you often use when we are addressing some of the diseases which we are coming across nowadays. So as far as emerging diseases are concerned, there are 3 ways a disease can emerge. The first one is agent changes itself. Maybe earlier it was low pathogenic in nature, and due to certain changes in its genetic makeup, it can become highly pathogenic. So that is one way of disease emergence by change in its pathogenic nature. The second one is previously some of the agents which have been there, but they were not causing disease of significant nature. But now they have been detected and they have been associated with a severe form of the disease. So that is one more way of looking at emerging diseases. And the third one is spread to new geographical area, can also be termed as emerging disease. So it can very well opt for transboundary emergence of a diseases if it is from country to country. And so these are a few ways we find the disease emergence. And I can give an example of porcine reproductive and respiratory syndrome virus, which was earlier located only to northeastern India, and now it has spread to rest of India in recent periods. And previously unrecognized virus, which was not there, but now we got it recognized. SARS-CoV-2 can be one of the emerging diseases. And avian influenza, we have low pathogenic avian influenza viruses, and which undergo a faster rate of genetic shift and drift. So that may give rise to kind of pathogenic viruses. And this is one more way how the agent or the disease emerges. So with this in background, now we'll get into the epidemiology of COVID-19. And I have arranged in such a way that trying to mimic the field scenario, because many a times when you come across the literature or the way we study at the classroom level, in order to make it better understand, we organize the things without considering the sequence of events. But here I try to bring in the sequence of events which likely to happen in the field scenario. So when you talk of any disease emergence, it starts with the case presentation. When I say case presentation, there is a clinician. If you talk about us like veterinary doctors, we have a veterinary clinician who is working in veterinary dispensary, and he is the first person who comes in contact with any disease. It may be an endemic routine disease, or it may be an emerging new disease. He is the person who comes in contact with the disease, and the case presentation, which means which is the animal affected, what are the clinical signs, what is the severity, which is the location where the disease has been found, and how far the disease has been spread. So these are a few questions as soon as a case is observed has to be considered in order to see whether is it a case of a new disease, an exotic disease, or an emerging disease, or it is an endemic disease due to not enough intervention. It has been occurring seasonally. The second point is why these cases are significantly important as an emerging infection. So any case presentation may not be that significant in terms of disease emergence. So when you consider a case presentation for its significance as far as an emergence of a new disease is concerned, that has to be considered into all those questions which will make it a unique case presentation as against already existing list of diseases. Then once we know the probably it is a case of a situation of a disease emergence, then we will have to know the causative agent, and if it is a bacteria, virus, or whatever the type of causative agent it is, then how it transmits. These are very, very important if you want to bring in the intervention strategies, and what are all the factors that are involved in disease pathogenesis. Again, finding out these factors are very, very important if you are planning for effective control and prevention measures. And what is the clinical manifestation, whether the disease is restricted to respiratory system, whether the disease is restricted to gastrointestinal system, is it a systemic infection or localized infection? So these all details has to be looked into in order to ascertain the severity and the nature of the disease. Finally comes, how are we going to diagnose the disease? Is it only by clinical signs, what we have observed, or it has to be supported by a specific laboratory diagnosis? Then comes, how do you differentiate these diseases from similar entities? So there are always a list of diseases which are to be differentially diagnosed in order to make our disease diagnosis confirmatory in nature. Then once we have crossed this 8th step, then we have at least some tools to handle the outbreak situation, which comes to either in terms of treatment for an affected animal, or in terms of protecting the unexposed animals against the infection. So this is a kind of sequence which I'll follow for COVID-19, and let us see how the disease has evolved right from its first presentation until the disease has been finally diagnosed. In a sense, the agent has been identified. So let us go back to the time around the month of December 2019, when most of the Indians were not really worried about this particular disease. Somewhere in the news and newspapers, we used to hear that there is something called COVID which has started in China, and it is causing some kind of unusual pneumonia among the human subjects. And so these are the kind of information we used to read in either in social media or in newspapers or in TV. But we were never having that feel like what is exactly going on there and what is the situation, what is the gravity of the situation. So those were all unaware when we were in the month of December 2019. So 31st December 2019 is the day when officially China reported the ongoing outbreak of a pneumonia of unknown etiology among the a cluster of cases in Wuhan, Hubei province. And again, why time and again epidemiology becomes so important. Just to give some background, how could they able to detect this pneumonia of unknown etiology? As China has been facing with a number of emerging diseases. Earlier it was SARS, and earlier to that avian influenza. And because of all these emerging diseases, they have a very good system of surveillance. And because of this surveillance system, they have been collecting the samples and screening it for most of the known respiratory infection, which included influenza, SARS, MERS, and these kind of agents were screened periodically under the system of surveillance. So when they collected these samples, and the clinical features and clinical picture was more towards the respiratory form of an infection. We'll come to clinical signs of the disease later on, but as such, right now, what report we have as on 31st December is there are a cluster of human cases which are showing respiratory clinical signs. When they were tested, they were negative for most of these respiratory infections, and they termed it as pneumonia of unknown etiology. So on 1st January 2020, because again the field situation becomes very, very important in order to associate any factor with the disease. So as they found out, most of these cases which are being reported and which were found to be pneumonia of unknown etiology, they all originated from one geographical area. And that was, they were having a link with seafood wholesale market in that particular Wuhan place. Because these are they are emerging or they are coming from one geographical area, the immediate step, the health missionary in that particular province and country, because majority of the cases emerged from a particular area, they suspected probably it is something to do with handling of seafoods and other wildlife animals which are traded for food purposes. Probably this is something to do with the handling of either the seafood animals or the wild animals, which might have led to this kind of pneumonia of unknown etiology, because China is already experienced with so many other respiratory form of infections which are very new, and probably it did not take much of a time for them to associate these kind of settings possibly responsible for the emergence of new respiratory infections. So as soon as they found this, and immediately the action taken was to seal the market, and all disinfection and sanitization steps were initiated. And by 3rd January, the number of cases reported were 44. And all the 44 cases were having similar clinical signs. But again, the etiology is not. So when I said the case presentation, so here the case presentation is, we have a 44 cases of human subjects. They're all having respiratory infection. But under surveillance system, they have found that all of them are negative for already known diseases. So this becomes very, very important and interesting. Like if it is negative for most of the known agents, then what is the causative agent for this outbreak? Is it something new? What we know about it? Can we term it as a new virus which is emerging? So because of this background, probably this case presentation is unique. Unlike any other case presentations where we know the agent, probably it is to do with other factors like the season or minor mutations in the agent or things like that. But here, none of the agents what we knew already, which were exhibiting similar clinical signs, they're all negative. That's how this particular case becomes very, very important in terms of identifying an emerging disease. So later on, this is a lady, she's a lady who's been detected positive for the first time outside China. She was traveling to Thailand and she was tested for the clinical signs. She was exhibiting for respiratory form of the infection, and she was found positive. So at this point of time, there were many unanswered questions. What we knew so far is there are a number of cases, and they're all having a pneumonia kind of signs, and they have all emerged from one geographical area, which is a seafood and wildlife market. But what we don't know are many. What is the cause of this disease? And what is the severity of this disease? Because so far, as on 3rd January, we had only 44 cases. Based on those 44 cases, can we predict the magnitude of this disease? How much mortality it can cause? How much morbidity it can cause? How fast the disease can spread? Is it severe enough? What kind of measures to be taken? So these are all the questions which will start emerging in the mind of an clinician or an epidemiologist in order to curtail or intervene the possible damage that can happen. Then what is the source of infection? So once you are able to identify the cause of the disease, then where from it has come? What is the source of infection? Then comes the nature of spread. How is it spreading? Is it close contact? Is it respiratory borne? And is it through aerosols or droplet infections or fecal-oral, whatnot? Since now, so far, the clinical signs were restricted only to respiratory system. So, what is the nature of spread it can cause? So, these are the questions which were automatically arise based on the information what we were having as on 3rd January. So, with that information, there need to be taken some kind of actions. Because we cannot wait until the disease is specifically diagnosed. We cannot wait until the source of the disease has been identified. Based on the information, we have to take an immediate action, which was done as a public health response by the Chinese government in that region of the country. Those were like the close contact have been identified. The close contact have been identified and were under medical observation. So there were total 44 clinical cases, and what they immediately did is they started doing a contact tracing. Now, by this time, everybody is familiar with what you mean by contact tracing, to define it. But once you know the clinical case of a disease, then people also have contact with the positive case. So those need to be tested, so that we capture all possible cases in a period of time, so that the spread of the disease can be curtailed. Then follow-up of close contact is ongoing. So one is the clinical cases. The second one is close contacts, what we have as far as the clinical cases are concerned. And those cases has to be observed for their exhibition of the disease signs. So that follow-up step is already taken. So this is a usual standard protocol what we do with most of the outbreaks when we try to investigate and recommend certain measures. Then comes the pathogen identification and the tracing of the cause, which was underway in that particular point of time. So during June 3, where we are now, so during that time, these were the kind of actions which were planned, and still we don't know what is the pathogen responsible for this. Then comes the Wuhan Municipal Health Commission carried out an active case finding using case definition. In most of the time, whenever there is an outbreak, people who express severe clinical signs or people who are not comfortable with ongoing clinical signs are the one which were captured by the health system. Otherwise, if it is a minor clinical signs or if the clinical signs are not disturbing day-to-day activities, most of the time they are not coming under the radar of the surveillance system. But in this situation, where we are suspecting the disease to be something new, capturing even those cases which were missed, just because they did not had that kind of need to go to the hospital and express their clinical signs and get tested, possibly for the cause what it was responsible for. Even now, in that scenario, it becomes a responsibility of health system to see that actively we go to that geographical area or a region and then identify our cases. How are we going to identify the cases? So in epidemiology, there is a term called case definition. So whenever we come across a series of cases with similar clinical signs, we take up those clinical signs which are most common to these cases and take them as a tool in order to identify the active cases which are prevailing in the population. This is very, very important in order to capture maximum number of people who have been exposed. Maybe the severity of the disease is different. But once we are able to capture such cases, we will be most effective in terms of curtailing the spread. So with that reason in mind, they have started the active case finding by this time. And at the same time, the disinfection and other sanitary practices and hygiene investigations were also carried out with those cases. So this is the public health response, which was done based on the observations done so far. So then comes the clinical diagnosis of a disease. So in any outbreak, immediate diagnosis is based on observing the clinical signs. So in this particular outbreak, the observed clinical signs were fever, cough, muscular soreness, and dyspnea. Further, when they were scanned and radiographed, they showed a kind of a glassy appearance in the lungs. There was a certain degree of consolidation in the lungs. There were atypical symptoms. In addition to what we have observed, a very common respiratory form of clinical signs, there were atypical symptoms like diarrhea, vomiting, and some of the acute cardiac injuries were also observed in the positive cases. And by this time, with a limited number of cases having been captured so far, the majority, in a sense, 25.2% of the patient were having some kind of underlying medical conditions. So these medical conditions were in the form of hypertension, in the form of diabetes, in the form of cardiac issues. So among the tested cases, this was a percentage with which there was an underlying medical condition. So with these clinical signs and the observed this one, they were further taken up for laboratory diagnosis. As I said in my initial slides, that because they were having a very good surveillance system, based on the clinical signs, the disease was suspected something to be related with the respiratory system. And immediately when it comes to respiratory system, these are the series of diseases which we know, which are there already, like influenza in general, which may be avian influenza and adenoviruses, and severe acute respiratory syndrome, which is the SARS coronavirus, and Middle East respiratory syndrome coronavirus. So when they screened these samples in laboratory, they were all negative for this. Hence the diagnosis was pneumonia of unknown etiology. So with this diagnosis, what we need to know further in order to make a specific diagnosis becomes a part of our epidemiological investigation. So risk assessment is very, very important to link between a source of a causative agent and the disease outbreak. As it was pointed out, the reported link to wholesale fish and live animal market could indicate an exposure link to animals. So that was the initial thinking which comes into mind based on the observations. And so far, there were no infection among the healthcare workers. And there were no clear evidences of human-to-human transmission. So, which suggests that there is a single source of infection, probably, as of now, probably from this particular market, wholesale fish or live animal market. It looks so far the disease, the spread is dead end. Like the 44 cases what we have observed, they got exposed with a particular thing in that market, and they are suffering the disease. So far, there is no evidences of spread of a disease from human to human. I remind you, we are still in January 3rd. But if you come to the June, we know so much. But I am trying to show you how the investigation started, how it progressed, what were the questions asked at different phase of investigation, how they were answered, and how the disease investigation progressed finally to identify what we know now. It is SARS-CoV-2, which was responsible for COVID-19. So far, so we could able to see that there were no human-to-human transmission, evident human-to-human transmission. And the symptoms reported among the patients were common to respiratory diseases. And it was a winter season again. Winter season, we know that respiratory infections are very common. Though it is a winter season and people are already susceptible and see environmental factors are favorable for respiratory form of infection. But still, it was negative for most of the known diseases. And the occurrence of 44 cases of pneumonia requires hospitalization clustered in space and time. So this space time is so important because all the cases emerge during that time. And all the cases have a link of wholesale fish or live animal market. So these two factors, space and time, makes it very, very important in order to further lead our investigation towards identifying the source as well as cause of the infection. Finally, on 7th January 2020, the researchers were successful in isolating the virus. Later on 12th January, whole genome sequencing was done. So once the whole genomic sequencing was done, then as you all know that molecular epidemiology, where we are trying to compare the agents at the genetic level, will help us to know the similarity or dissimilarity between the genomes, will help us to know where this particular agent or where this particular virus evolved. So whole genome comparative analysis revealed that novel coronavirus resembles the bat coronavirus. By then, because see, it is very difficult to narrow down which are all the viruses we are going to compare, because there is already the understanding of emergence of MERS, emergence of SARS, and the clinical signs were known, the severity of the disease is understood to some extent. Based on that, we already suspect this particular infection to be originating from certain group of viruses. So with that understanding in mind, immediately the whole genome sequences of the current isolate was compared with the existing coronaviruses of all animal origin, but it was found very similar to bat coronaviruses. Later on, we'll come to like what percentage of similarity it was found with the bat coronaviruses and what was the extent of similarity found with the MERS and the SARS-CoV and all those things. But as of now, we know that the virus which is being suspected to be responsible for this particular outbreak is resembling bad coronavirus. And by this time, the number of cases have been increasing. I'm not getting into that details because till we identify an agent which is responsible for the infection, tracking the cases, and later on, now we have cases in lakhs and crores, and based on that understanding and latest information, we know that there are certain risk factors associated with the host, agent, and environment, which are contributing for the severity and the extent of mortality and morbidity, which we are observing in a different population across the world. So few of the risk factors, most of you know it already, like age is one important factor. We have been observing old age are more susceptible and the severity of the disease observed more in old age group. And comorbidity is also known, like hypertension, diabetes mellitus, asthma, cardiac issues, you name, there are a number of underlying conditions which make an individual susceptible for infection. Gender, you may be knowing or may not be, male are found to be more affected than females. Even the latest research says that but the receptor which is required for a virus to attach and enter into the cell, the distribution of that receptor is gender specific. And males are having more distribution as compared to female. So this has been established, and a recent paper has been published in that line, and a similar trend has been observed across countries. Race and ethnicity is one more observation, which is like African Americans were found to be more susceptible and as compared to Native Americans. And some medical conditions, as I said, respiratory chronic forms like kidney malfunction, etc. Use of certain medications. You know that there are a number of drugs which can cause immunosuppression. And now it is a very good understanding among most of us, like if your immune system is, if your general immune system is strong enough, probably we can handle this infection very well. But if your immune system is compromised, probably you are a victim to the infection. And other factors like poverty and crowding, which will indirectly affect the other factors of the host susceptibility. Pregnancy, we know that physiologically the pregnancy status is known to have a little immunocompromised state and probably the susceptibility. So these are few factors which are risk factors so far enlisted, which are associated mostly with the host, which is a human subject, and some of the micro environment conditions like poverty and crowding. Those are the things. So these are a few of the risk factors which we know so far, which can affect the individual to a different extent. So coming to a second Mythbusters, as I told, every now and then I have some of these Mythbusters which will convey a certain message. Most of you might be knowing, or some you may not be. Can shoes spread the COVID-19 virus? And the answer is the risk is very low, because you know that it's a respiratory form of infection. The likelihood of COVID-19 being spread on shoes and infecting individuals is very, very low. But as a precautionary measures, particularly in home where infants and small children crawl or play on floors, consider leaving your shoes at the entrance of your home. This will help to prevent contact with the dirt or any waste that could be carried on the soles of the shoes. So this is one more commonly observed phenomena. And coming to the disease transmission, so far we try to look at what was the initial case presentation, what were the epidemiological observation when the cases were reported, and what was the causative agent, and what were the host risk factors that will decide the severity of a disease. So now comes the disease transmission. So until 3rd January, we were of the understanding that probably human-to-human transmission is not there. But later on, we observed, and there is established figures to show that there is an effective transmission of infection from human to human by all means. So which are all, depicted here. Based on the sequence comparison, now we know that the coronavirus might have emerged from the reservoir host, which is a bat so far, because based on the similarity, we found that it is very close to bat viruses. And then most of the time, so far, the viruses, the human viruses that are emerged from bat reservoirs always had one intermediate host, which is in the vicinity of human habitation. So far, as far as SARS-CoV-2 is concerned, we don't have an identified and confirmed intermediary host. So that is a very good point of research which has to be looked into. We don't know yet which is an intermediate host between the bat and a human, which is efficiently transmitting the disease. Transmission from bat to human directly is a less chance because of the settings what we have. And so far the suspected intermediate host, which might be pig, wild birds, chicken, cat, bat, dog. Pangolin is one more animal which has been found to have a coronaviruses which are similar to SARS-CoV-2. That is how it is also been suspected to be one of the intermediate hosts. And other animals like horse, camel, cattle, goat, and sheep also have a set of their own coronaviruses, but we don't know yet what is their role in the emergence of current SARS-CoV-2. And cross-species transmission, as it is being said, as of now, pangolin is the one which is plugging the cycle, and it has been considered as one of the intermediate hosts, but still we are not very clear which is the real intermediate host. So disease can transmit from such intermediate host to human by zoonotic transmission. Once it is human is infected, that is the first event which has happened in initial cases. Later on, it is human-to-human transmission. It can be through community contact, or the hospitalized individuals can transmit it to nosocomial healthcare workers, or it can also transmit to other patients which are in the hospital. And this is the immediate means of transmission. But if it has to spread across a larger geographical area, then probably the infected individual should have to travel, and which we have witnessed now. We all know that the disease from China to the rest of the world got transmitted through the movement and travel of infected individuals across the continents. And this is the means by which disease is transmitted. It is the close contact between the susceptible and the infected individuals which led to transmission. So one is the disease transmission. The second one is rate of transmission. So every disease, in order to propagate itself in a population, it has to spread from an infected individual to a susceptible individual. So that cycle has to continue so that the virus persists, propagates, and remains in the system. The second one is how fast it is able to transmit from one individual to another. So that is measured by a very important parameter, epidemiological parameter, which is called R0. Again, with the kind of information we have in social networking, probably you already know it, like what you mean by R0. It's a basic reproduction number, which is used to measure the transmission potential of a disease. How fast and how efficiently the disease can transmit is measured by R0. And on your left, you see like the series of respiratory-borne infections and their R0 value. For COVID-19, initially it was 2 to 4. But now it is between 2 to 2.5, which means an infected person can transmit effectively 2 to 3 people if they are susceptible. And again, the R0 concept works in a population which is susceptible. So if the population is susceptible, and if there is an infected individual, how efficiently he can transmit will tell us the rate of transmission. And COVID-19 is 2 to 2.5. When it comes to SARS, it was 2 to 4. When it comes to MERS, it was 2.5 to 7.2. The higher the R naught value, more faster is the spread of a disease in a given unit of time. So that's how our effort as an epidemiologist, our effort as an policymaker, our effort as an administrator is to see that what intervention can reduce this or not value. And you're all familiar now, and you have been experienced the total lockdown period. Now we are also experiencing the partial lockdown periods. The entire effort of lockdown was towards reducing the rate of spread, which is towards reducing the R naught value. That is one of the reasons why the lockdown situation or lockdown measures were planned. There are many other factors which are important and which are associated with the lockdown in order to stop the spread and also minimize the disease in the population. And R naught is influenced by some of the factors. One is how frequently the infected person comes in contact with the susceptible individuals or susceptible population, and what is the probability of an infection being transmitted during the contact. So at each level there is certain probability. One is the rate of contact between the host population. Second one is the probability of infection being transmitted during the contact. And the third one is the infectiousness of an individual who has been infected. So these are a few very important factors which will decide whether your value ranges between 2 to 2.5 or it may be higher or lower. So based on these factors, the rate of transmission can be affected. So now just to summarize so far what we have done, if I have to take a lecture on outbreak investigation for a BVSC students, probably I'll start in a way what I have enlisted here. So this is the methodical way of understanding what is the outbreak investigation, because outbreak investigation is the first epidemiological activity which we are going to do in any disease emergence. So as I started referring to the emergence of 44 cases and epidemiological settings, and later on how the things progressed, that phenomena, those steps can be brought into some general steps which are very fundamental for any outbreak investigation for that matter, not necessarily the COVID-19. In order to do an outbreak investigation, first you have to confirm whether it is an outbreak. Again, it comes to the definition of what you mean by outbreak. So outbreak definition, again, it changes with the disease. If there is no disease and if there is even a single case, it is an outbreak for us. But there are endemic diseases which are there already in the population. Say, for example, FMD. There is always a certain number of FMD cases which we are observing across a geographical area in a given country. If the number of cases goes beyond those observed usual number of cases, then it is a case of, then it is a situation of an outbreak. To make it more objective, like there are 10 cases observed on an average during the month of June or July every year. And this June you are finding around 100 cases. Then it is a case of outbreak. But as far as SARS-CoV-2 or COVID-19 is concerned, even appearance of one case is an outbreak for us because the disease was not existing and emergence of one case is also an outbreak. So first of all, confirming an outbreak even before you start investigation is very, very important. Whether is it a regular phenomenon in the field or is it a special case where we need to be investigated so that something has changed in the risk factors associated with the environment, host or an agent which might have led to such an increase in cases. So these are the certain things, steps which then once your outbreak is confirmed, then you have to verify diagnosis. So this diagnosis verification is done through clinical diagnosis, laboratory diagnosis, using different methodologies. Then, as I also referred, the case definition and identification of the cases, which I referred when I was talking to you about active case identification, which is done based on the case definition. Then we do certain descriptive epidemiology, like what is the total number of cases observed in a population, what is the morbidity, which means how many total number of cases are there in a population, what is the mortality of the affected, how many died, what is the case fatality. So these are a few parameters which we have to be familiar with, and most of you might be knowing it. Then comes the data analysis, and finally creating a hypothesis to say that these are the probable factors which might have led to outbreak. Then those hypothesized factors has to be researched to identify whether they are really the factors which we think so. And finally, our control prevention measures has to be placed in. Because entire 6 steps are finally to lead us towards effective control and prevention. So this is the part which we covered right from our first slide to the latest one. And again, just to put it in a more perspective, as I said, if it is a case of unknown etiology for COVID-19, it started with cases of unknown etiology, the 44 cases. Once you have such cases, we are already facing an outbreak. So based on this situation, what the epidemiologists and clinicians did is they observed the clinical signs. And based on the clinical signs, they found it is as a case of a pneumonia. And pneumonia is a systemic diagnosis. And you know that if it is a pneumonia, what kind of treatment to be given. And once you know the clinical signs, you have a tentative diagnosis based on which you start your intervention process, which may be a treatment. And based on the field observation, you also know the risk factors, which will also further help us to target the control and preventive measures. Again, taking the example of COVID-19, the risk factor, what they found in the initial observation was exposure to seafood and wildlife animal products. Because they found that to be one of the risk factors, they immediately took an intervention strategy of closing that market, thinking that probably the disease will not spread further. So that was how the epidemiological factors identifying during the investigation will help you to do the control and preventive measures. And further, so still we haven't diagnosed the disease specifically, and entire observation and the steps were taken based on the clinical signs observed and the risk factors identified. Then comes to lab confirmation. As I pointed out, the virus was isolated and it was sequenced. Once it was sequenced, we know that it is a case of SARS-CoV-2, which is a new virus which has emerged probably from that. And now we have much more specific information as far as outbreak is concerned. And now you are having much more specific control and preventive measures. This entire process which I am depicting here again goes with your steps of outbreak investigation, and finally our aim is to control and prevent the infection. So with that in background, again coming to our Mythbuster 3, the prolonged use of medical mask when properly worn does not cause carbon dioxide intoxication nor oxygen deficiency. Probably most of you might have experienced it already, like when you use a prolonged thing, and it is uncomfortable, and but it doesn't really lead to the biochemical definition of carbon dioxide intoxication, but it's only lack of breath and lack of air. Probably that will be taken care. And while using a medical mask, make sure it fits properly, and that is tight enough to allow you to breathe normally, and do not use a disposable mask, and always change it as soon as it gets damp. So as of now, the scenario of COVID-19 worldwide, if you look at, it's the latest data which I took it from WHO website, and the number of cases, confirmed cases, and these are the number of deaths so far observed. And if you look at the confirmed cases, it is still an increasing trend as you start from January 31st, and you see the progression of the case, and it is still in an increasing phase, which means to say the epidemic is still, if you consider the global scenario as such, country-wise, there may be a declining trend, but if you consider the overall worldwide cases, probably it is an increasing trend, and India is contributing significantly for these active cases which have been reported lately by WHO, and a number of deaths also by and large it is maintaining. As we also, if you look at our Indian scenario, the total number of samples tested so far are around a crore, and active cases were around 2 lakh odd, and cured discharged cases are around 4 lakh, and deaths so far are around 20,000. And highest cases were reported from Maharashtra, and the lowest, it is the lowest cases, which is from Lakshadweep. I just correct here, it is the lowest cases, which is from Lakshadweep. So this is in brief about epidemiology of COVID-19, and those are the kind of concept which I wanted to discuss. And then comes the SARS-CoV-2 and its evolution. So by the end of our epidemiological investigation, we know which is the agent responsible for the infection. And now we are trying to understand further how this virus evolved, and is there any other similar viruses in livestock or in wildlife or in other species of animals. So if you look at SARS-CoV-2, the classification of the viruses, it falls to order Nidovirinae and family Coronavirinae, subfamily Coronavirinae, and genus is beta coronaviruses, and group B. And virion is a single-stranded positive sense RNA virus, close to 26 kb genome size, and it's an enveloped virus, and spherical shape around size 100 nanometer. And if you look at the virus, it has structural proteins and non-structural proteins. One of the protein which is very, very important for our understanding is a spike protein. And the spike protein has significance in terms of identifying the target cell in the infected host. And it also has a significance in terms of producing the neutralizing antibodies in order to neutralize the viral infection. So because the arrangement of the spike protein is such that it gives an appearance of crown, and that is how the name has emerged as coronavirus. The other important proteins include nucleocapsid proteins, envelope proteins, and membrane proteins. But we'll come to the spike protein later in order to understand its significance in terms of disease. So if you look at taxonomy of coronaviruses, as pointed out, among Coronaviridae family, there are two subfamilies, Coronavirinae and Torovirinae. And Coronavirinae is the one which has genuses alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. Most of the animal viruses, they are under alpha coronaviruses, and there are few beta coronaviruses, and it is the one genus which hosts the human coronaviruses and some of the bat coronaviruses, and the gamma coronavirus and delta coronavirus also has some of the viruses of bird origin and rodent type. And to draw your attention, except the human, most of these coronaviruses in animals predominantly have clinical signs associated with the gastrointestinal tract, which means the prominent sign is diarrhea and associated with that. Having said, they also have a respiratory infection, but it is not as severe, as predominant as we observed in case of human. So the phylogenetic tree of coronaviruses. Phylogenetic tree is again a term which is often used in order to compare the sequences of a virus or a protein or a gene in order to establish how close, how similar or dissimilar is the virus. So the protein or the gene targeted was RNA-dependent RNA polymerase, and around 50 virus sequences were compared in order to see how the grouping happens. And as said already, SARS coronavirus 2 comes under group beta coronavirus, which is mainly a bad, which has mainly a bad coronaviruses. And as I said, the beta coronaviruses are subgrouped into A, B, C, and D. And it is beta coronavirus B, which has our SARS coronaviruses, whereas beta coronavirus C has our MERS coronavirus. And in addition to, so beta coronavirus B and beta coronavirus C both have a group of bad coronaviruses. And when you come to the comparison between the viruses of bat origin, pangolin origin, and civet origin, so red dots are the one which are sequences from human subjects, and the blue ones are from the bat origin, and yellow ones are from the pangolin. Pangolin is just a scaly anteater. The name is, it's a synonym in a way. And as I was pointing out, 98% identity was found with one of the bat coronavirus, which is RATG13. So this is the highest identity percentage which we observed, and it is with the bat coronavirus. Next, again, 88% of the identity was found with two more bat coronaviruses, whereas it was 79% identity with our SARS coronavirus and 50% identity with the MERS coronavirus. And the coronavirus sequences in pangolin was also having a certain similarity. So among the species of animals which we compared, human, bat, pangolin, and civet, so far the coronaviruses detected from pangolin and bat are having certain degree of similarity with the human coronaviruses. That is how still we are not in a position to specifically say that which is a real intermediate host between bat and human. And based on that information, the proposed the evolution, or in a way the transmission of the virus might include the direct transmission from bat to human is less likely for this one, and the transmission from corona to pangolin, on pangolin to human, is one probable route. And this question mark indicates that there might be any other intermediate host, which we don't know as of now, which might be transmitting the coronavirus, which has been contracted from bat to human. So far, this is the understanding what we have as far as role of animal as an intermediate host in transmission of, in evolution, rather emergence of this particular virus from bat. Again, the Mythbuster 4, most people who get COVID-19 recover from it. At least as we know now, in India, majority are getting recovered unless there is an underlying factor of age or any comorbid condition. But it is not very general to say like that. Most people who get COVID-19 have mild or moderate symptoms and can recover based on the supportive care they get it. As far as virus replication is concerned, this particular virus has a number of target cells and organs in the body. You might be aware, the latest research says almost every organ is getting infected by this particular virus. Only the damage and the changes what we observe varies between the organ systems. So initially, being a respiratory form of infection, the infection was the target cells were lung cells. In lung cells, it is the epithelial cells, type 2 pneumocytes, or it may also be alveolar macrophages, which can be a target cell for SARS-CoV-2. Then comes gut. In general, we know that coronaviruses can infect both respiratory as well as gastrointestinal tract. Only the severity may change as usual. And kidney, liver, pancreas, and heart are the other target organs which have been infected by this particular virus. And this virus and also other coronaviruses have a specific receptors on the target cells. So distribution of these receptors will decide whether that particular cell or that particular animal is susceptible for this particular virus infection. So far, what we know is ACE2 is one particular receptor which is abbreviated for angiotensin converting enzyme 2. With this particular receptor is present in most of these target cells which I enlisted below. And the interacting pattern for this particular receptor is S protein. As I was referring while I was talking about the virus structure, S protein is very, very important. So this has two domains, S1 and S2 domain. S1 domain is the one which interacts with the receptor of the virus and receptor which is present on the target cells. And that interaction will facilitate the endocytosis of the virus into the target cell. And replication of the virus takes place in cytoplasm. And because it is a positive sense RNA, replication immediately starts with RNA-dependent RNA polymerase enzyme, which is virus associated. And once replicated, the virus is released out through the infected cell and by exocytosis. And these cells will further infect the cells which are at vicinity. And similarly, if you look at other coronaviruses, the receptors are like aminopeptidase N, which is abbreviated as CD13 for transmissible gastroenteritis virus, PEDV, porcine epidemic diarrheal virus. So these are kind of coronaviruses which are important for animals and especially for the pig. And MERS coronavirus utilizes dipeptidyl peptidase 4 as a receptor for its entry into the target cell. For time being, for us, ACE2 is very, very important in terms of SARS-CoV-2 receptor and its infection. So this was a very popular myth among most of the people, and it was circulating for quite some time, and they started equating like since sanitizer also has alcohol, drinking alcohol will protect against COVID-19, which is really a myth. And you know that the harmful effects of the alcohol can make you susceptible for COVID-19. So don't be under the concept like alcohol drinking will make you protected against COVID-19. So that is about the virus and how it was detected and what similarity it showed with the other viruses in different animals. And based on that similarity, what we know in terms of disease emergence. Now we'll get into animal coronaviruses, which are a number of animal coronaviruses are there. Let us start with COVID-19 as such, because initially there were reports of COVID-19 causative agent being detected in pet animals like dogs and cats. Later on in wild animals, captive wild animals, in tiger, lions, and mink, which is a semi-aquatic kind of animal. But so far, there are no evidences to prove that the infection can transmit from dog, cat, or any other animals to human. And in all these reports, the epidemiological evidence was these animals were in contact with infected human being. So, which means to say the infection is, the zoonosis is reverse zoonosis. It is from human to animal in these situations, but not from animals to human. So far, our understanding says that disease can transmit from infected human being. If he comes in close contact with their pets or any other animals, it can transmit. So having transmitted, so far there were no clinical signs observed in dog cases, whereas cat, tiger and lion and mink, there were very degrees of respiratory or gastrointestinal illnesses, but they were not severe enough to be kind of alarming for the animals. So based on this and earlier reports also, if you look at it, there is always an intermediate host for most of the bat or rodent-borne infections, viruses, to finally end up in human being. So there is a human coronavirus 229E, which llama was an intermediate host. And there is human coronavirus OC443, bovine was an intermediate host. And SARS coronavirus civet palm civet was found to be intermediate host. As MERS coronavirus, we know that already camel was an intermediate host, and especially dromedary camels were the one. And there is one more coronavirus which has been observed, and pig was suspected to be one of the intermediate hosts. So this way, the livestock are playing a significant role as an intermediate host in transmission of these diseases or in evolution of coronaviruses. Similarly, if you look at this diagram here, 2019 N coronaviruses, still it's a big question mark, which are all the animals probably doing the intermediate job of an intermediate host, which is not very, very clear. Coming to animal coronaviruses, as I said, most of the animal coronaviruses comes under the genus alpha coronaviruses. And if you look at it, there are feline coronaviruses, canine coronaviruses, and transmissible gastroenteritis virus of the swine, and porcine respiratory coronaviruses. And so these viruses, if you look at the second column, which is a disease and the symptoms, most of them, they tend to exhibit gastrointestinal clinical signs, except for feline, where the peritonitis was predominantly observed. And transmission is by fecal-oral route. And some of these animal coronaviruses do have vaccines, which have been available locally, if not in India. And as far as one of the viruses in pig, which is porcine respiratory coronavirus, which is a kind of deletion virus from porcine epidemic diarrheal virus, which causes a mild respiratory disease, and which is persistent virus in case of pig. And we are looking into these viruses and their prevalence in the amidst the outbreak of this particular COVID-19. And as they are, it is having a respiratory form of a disease. Transmission is by aerosol. And since the disease is not causing any damage to the animal, there are no such vaccines or preventive measures which are there. The next one is porcine epidemic diarrhea, which is a very, very important disease, and it's an emerging pig coronavirus disease. So far, not reported much in India, but this disease is very, very important, and it can cause high mortality in piglets as well as even in affected sows. And transmission is by fecal-oral route. And this is one more important coronavirus which is observed in porcine. In addition to the alpha coronaviruses, there are a few beta coronaviruses. Again, pig has one, which is a porcine hemagglutinating encephalitis virus, and mouse and rat are having few. And bovine coronavirus is one more important virus, which is associated with the gastroenteritis, gastrointestinal symptoms, and cough diarrhea. Or cough diarrhea is a complex of agents, and one of the causative agent is bovine coronavirus. And it is very, very important and economically important in terms of feedlot cattle. And the transmission is by fecal-oral route. And there are equine respiratory coronaviruses and canine respiratory coronaviruses, but their significance is not much. Then comes group B. And among, as I said, beta coronavirus is divided into group A, B, C, and D. A has most of these viruses, whereas B is the one which has your SARS coronavirus, which is a kind of zoonotic emergence originating from bat, and transmission is aerosol and oronasal secretions. So far, there are no vaccines available, but due to effective biosecurity measures, the disease was controlled. And comes your gamma coronaviruses and delta coronaviruses. Among gamma coronaviruses, it's mostly the birds, avian as well as turkey. Whereas delta coronaviruses, it is porcine delta coronavirus is the one which is again important in terms of its economic significance. And other viruses are there which are not so important economically for livestock, but they fall into a group of coronaviruses. So this is to realize that there are a number of coronaviruses falling into different genuses in which are affecting the livestock. What is the role of these livestock in emergence of new coronaviruses? As we have seen, the bat coronaviruses giving rise to a SARS-CoV, SARS-CoV-2, MERS, and other kind of emerging infectious diseases. We don't know yet like what these livestock animals can play. So far, there are no such strong evidences, except for MERS, where we have a camel as an intermediate host. Similarly, other livestock can play a role in the emergence of new diseases. So this again, does housefly transmit COVID-19? And as of now, there are no evidences or information to suggest COVID-19 virus and transmitted by houseflies. And possibility is because so far we know that this COVID virus transmission is either aerosol or droplet infection. Probably such a droplet infection might be transmitted mechanically by this one housefly. But such a transmission, how efficient it is and how far it can cause a disease is yet to be researched. We don't have any evidences to say that. So final section of my talk will include what are the control and preventive measures so far. Most of you are very, very familiar. And next one is biosafety guidelines, which is very, very important. Now onwards, most of the universities, research institutes are planning to take up a research activity associated with the COVID agent. Probably we have to have a fair idea about what are the biosafety guidelines to be followed. And then comes the COVID-19 research priorities in animal sciences as recommended by OIE. Soon after this outbreak, OIE called specialists across FAO, WHO, OIE, and other agencies, and they have formulated a document to guide the future research in COVID-19 and the priority areas. In line with that, ICAR also has taken certain initiative to do a kind of research projects, a network research project on COVID-19, which is still in a preliminary stage, but the discussions are on. And finally, conclusion of this particular talk. So as you know that at individual level, we can maintain a social distancing, wear a mask, hand wash using a soap, hand sanitization using alcohol-based sanitizer, which is pretty much known to everyone as of now. And social distancing of 2 meters is what is recommended. And community level spread can be stopped only by stopping the movement of people, which is possible only through total lockdown or partial lockdown, either nationally or regionally. The third option, what we have is improving our general immunity. That is the thing which one can play a role, because as of now, we don't have a vaccine. So only way we know that the information says that people with the general health condition, general immunity, are able to better handle the infection of COVID. So exercise and yoga, even WHO recommends doing exercise, yoga can help your health to maintain the immunity, and nutrients to boost your immunity, good nutrients to maintain. And other practices like most disinfect the high touch surfaces and use your use tissue paper or kerchief or your hands in order to, whenever you sneeze or do things, and clean the area properly, and try to avoid the movement either locally or this one as far as possible. And used mask has to be disposed properly. Nowadays we are seeing already on roadside, you are finding number of masks thrown here and there, and we don't know how many of them are infected. So that kind of civic sense has to be there in order to prevent possible transmission. And no need to say much how to use a mask. You already know that your hand should be clean when you are touching your mask, and you decide which is the top side and which one is the side towards your nose, and the color difference as well as the presence of that nose band will tell you which side you have to wear, and place it on you and press it along the nose so that it fits well, and you are protected against possible droplet infection. And once you discard, ensure that it is discarded safely, and soon after discarding, you wash your hand with the soap. And frequent touching the mask, touching the outer surface of the mask, and removing the mask and talking to the people. And these are all certain observations you might have already come across. You know what for the mask is worn, and if you deviate from the purpose for which it is wear, then probably you are not well protected. Coming to biosafety guidelines for COVID-19 diagnosis and research, as of now, the agent is been categorized into risk group 3. You know that all the biohazard agents or infectious agent has been categorized into four groups: risk group 1, risk group 2, and risk group 3 and 4. And based on risk grouping, we know what kind of biosafety laboratory we should be using. BSL-2 is recommended for non-propagative diagnostic work, which means you are collecting the samples, screening it. Only to that extent you can use a BSL-2 facility. When it comes to virus culture, antigen preparation, so those kind of activities where you are propagating the virus, you should do under BSL-3 facility. And packaging of materials recommended is triple layer packaging. You all know that how we pack the anthrax samples. Similarly, it is the recommended practice for COVID-19 suspected samples. And transportation should obey the UN 3373 code as well as UN 2814 code, depend upon whether you are sending only the clinical samples for screening or you are sending the viral culture for further analysis. So these are very basic biosafety guidelines which I brought to your notice. There are furthermore details. What you mean by BSL-2 practices, what you mean by BSL-3 practices. There are very detailed DBT documents to support you in this line. And spraying or introducing bleach or another disinfectant to your body will not protect you against COVID-19 and it can be dangerous. So we came across instances where people have been sprayed with the bleach, which it is not safe. As you know that bleach is a very corrosive in nature. It can cause your sensitive organs like ear and other mucosal surfaces. So one should not spray yourself with bleach or any other disinfectant. And coming to the fag end of this presentation, as I said, COVID-19 research priorities in animal sciences are being already charted out. Surveillance and risk assessment is one important area. As I said, unless we do a surveillance, we don't know what all livestock are involved in emergence of.
Hello? Sorry to disturb, Dr. Jagadish.
Yeah.
You have last 5 minutes to sum up the things.
Last proposal. Yeah.
Thank you.
Yeah. So surveillance and risk assessment is one thing, and identifying the animal reservoir and intermediate host through surveillance and investigation strategies, which considers the transmission pathway also. And host range, what is the host range? So far we know that it is being transmitted from human to other animals and wild animals. We don't know other range of species which can be infected. And dynamics of wildlife trade, how it is affecting the COVID. Or any other emergence of such new viruses, and possible role of livestock, we don't know yet. Possible role of companion animals in epidemiology of human diseases. If your pet animals are possible to get infected with these viruses down the line, what will be the role of these pet animals in terms of virus evolution? And what we need as a tools in order to control or prevent diseases, diagnostics, vaccine. And this is one more area which is very, very important. And then comes our socioeconomic impacts and the policy things, which is very important in terms of administration. And again, exposing yourself to sun or temperature will not really protect much against COVID, because you know that it can survive as long as it can survive in your droplet infection. So if they are dead, either there is temperature or not. So that is one more thing to be looked into. Coming to the ICR Research Institute in COVID-19, as I said, ICR is providing COVID diagnostic services so far. The initial service, what we are doing as an ICR Institute is we are providing diagnostic service for human. So 3 of our labs are already into the process, and most of you might be knowing it. And then comes the network project on COVID-19 is under consideration, and it is in preliminary discussion with 3 theme areas. One is epidemiology of coronaviruses, and diagnostics for coronaviruses, and vaccine for coronavirus. With these 3 theme areas, a network project on COVID-19 is in formulation so far. And this is one more thing which it was a little interesting, like being able to hold your breath for 10 seconds or more without coughing or feeling discomfort does not mean you are free from the coronavirus disease or any other lung disease. Usually we tend to make such attempts in order to see, and people tend to say that if your lungs are healthy, you can hold your breath. But probably this is not a right way to test whether you are susceptible or not for this particular disease. So in conclusion, COVID-19 is one of the biggest pandemic witnessed across the globe so far, and bad coronaviruses have been the source of recently emerged SARS coronaviruses, not necessarily the current one, the previous one also. The intermediate host for SARS coronavirus 2 is yet to be identified. So far, we know it's pangolin, but we don't know yet which is the real one. Animals play a critical role in evolution of human coronaviruses, which need to be further investigated. And finally, dryers, we know that it is not enough. It is not enough to kill viruses, so it is not done. And these are few references. I can give a few more. And so age-based susceptibility is already known. And thank you so much for your kind attention so far.
So that was an excellent presentation, exceptionally crystal clear in explaining the different aspects of the epidemiology of COVID-19 and other coronaviruses in man and animals. Thank you very much, Dr. Jagadish. The forum is open for discussion, and I will read out some of the questions posed by the participants. The first question by Dr. Nirupama Jaising. Our question is, what is the other mode of transmission of COVID-19 other than aerosol route? Can it be transmitted by material and food?
Yeah. So far what we know is aerosol transmission is a direct transmission between an infected individual to a susceptible individual. The second one is, as it comes to material, we already know that if a surface is infected due to the presence of droplet, and if you come in contact with such surfaces and you happen to take such surfaces to either in the form of breathe or in the form of this one, this is one more way you can get the infection. That is how frequently it is recommended that the surfaces which you regularly come in contact, clean them, sanitize them. That is one indirect way of getting the infection. The direct way is if the susceptible individual your vicinity, and if he sneezes, if he coughs, or even otherwise, there is a chance of aerosol transmission which directly can be infected. Through food, as of now, there are no such evidences to say that through food there is a disease transmission, because food undergoes certain processes, and to be state like there is no such evidences to say that. Hello. Hello. Is it audible?
It is audible.
Yeah.
Let us take the next question from the chat box.
Yes.
Yeah.
I think there is some issue.
What is the effectiveness of the N95 mask? This is the question asked by Dr. Sripagarav.
Yeah. So, as such, the recommended PPE practices. So one of the important recommended PPE practices is your mask. N95 effectiveness is effective because it already says N95. 95% of the agents are filtered, and it is been recommended for people who are in high risk zone and people who are directly handling the things. And if he has to say, like in terms of is it effective or not effective, yes, it is effective. And in terms already the mask says how far it is effective.
We expect a very brief answer because we are about to terminate because there is time.
Okay, please go ahead.
So very very brief answer because there are many questions that need to be answered.
Okay.
Please,
Okay.
please go ahead.
Yeah, N95
Upon passage of time. Yeah, please.
Done. Please go to the next question.
Yeah. Mr. Deepesh Sharma is asking a question upon passage of time whether the virus loses its pathogenicity.
Say it again.
Upon passage of time, whether the virus is going to lose its pathogenicity.
It might, because we know it already, this is an RNA virus. Both are possible. It may lose its pathogenicity depend upon the type of mutation it accumulates, or it may also change to become a little more virulent virus. So we don't know how it evolves.
Okay. Nandini is asking the question about the nervous system involvement. Do these youngsters, they are asymptomatic?
As of now, the data says that the significant population is asymptomatic, and already we have come across such data. And yes, there are a good number of population which is asymptomatic.
Hello, Dr. Nagappa.
Sunday Arani is asking the question. Yes, sir, please. Some questions are already asked. You please start. Continue, please.
Hello.
Yeah.
Hello. Can you hear me, Dr. Jagadish? Yeah. So there was some signal drop.
Yes.
There's a question from Dr. Manjunath. So how long
Yes,
the
sir.
virus stays in the blood? Can we track the virus in the blood by RT-PCR? How long the virus stays in the blood? Can we track the virus in blood by RT-PCR?
Yes. Virus can be detected by RT-PCR through, in the blood, but the very good sample is our throat sample, throat swabs. But virus in the blood can also be detected. And there is a phase 1 during which the virus is available, and it is in a systemic circulation. It can be detected.
There's some question from Dr. Satyabrat Datta. Does herd immunity strategies are possible to defeat COVID-19 in our country?
So far, it looks like that is the only way we can defeat it, sir. There is no other way unless we get a vaccine done. Otherwise, there is no other way of handling it.
So there is one question from Dr. Mallinath.
Yes, sir.
If RO is less than SARS and MERS, why both have not become serious compared to COVID-19? He's asking direct relationship probably with RO, SARS, and MERS.
Yes, sir. That's very interesting in a sense, because the efficiency with which the virus is able to transmit from human to human, though the rate of transmission is less, but the efficiency of transmission is good compared to other two viruses. And that is how it is able to spread across population and across countries. The rate is one factor, but rate alone will not decide the spread of an infection. There are other factors which will contribute for the spread of an infection. In that sense, it is efficient.
Okay, thank you. We have this one more question from Dr. Datta. Does the coronavirus affect the sense of taste? I don't know whether this was asked or was. Does the coronavirus affect the sense of taste? Taste sense, yeah.
Taste sense, yeah. I'm not sure, sir, like whether it has anything to do with taste. Because in general, if any infection will have an effect on our tasting ability, but specifically if it has to do anything with our sensory system, that I'm not sure.
Even in the common cold, we lose that sense taste.
Yeah.
So, okay, that's thank you.
Yeah.
So, Dr. Abdul, can we have some more questions, or is it time for us to wrap the session? Dr. Abdul?
Yes, sir. 5 minutes left, sir.
Pardon me?
5 minutes left,
5 minutes, okay. So, I'll just find out a few more questions.
You can ask it.
Yeah, okay, right. I don't know, well, this question asked this, what is N95 mask? Can we use N95 mask repeatedly after daily wash? Over. Okay, so I was offline for that.
Yes.
And one more question, sir. What are the candidate genes of SARS that are being currently considered in the development of vaccines?
Yeah.
SARS
Spike protein.
that are being.
Spike protein is one which is targeted as far as diagnostics as well as vaccine is concerned.
So there are so many participants expressing that this presentation was very nice and thanking you.
Thank you, sir. Thank you.
Okay, so how long the virus stays in the blood? Okay, we asked this question. There is one question from my side to Dr. Jagadish. So regarding the role of bats, so how confident are we from the scientific community? Because there was a lot of miscommunication and people started attacking the habitats of the bats. So the bats are actually the farmers-friendly creatures. So this kind of misconception, so in the minds of people. So what is the solution, and what is your message on this?
If you look at it a larger context, why we are able to, have such emerging diseases, there are multiple factors to it. We are encroaching into so many habitats which we are not supposed to be there, and because of that, we are disturbing the natural setup which is balanced in the wild. And due to that, there is a possibility of such viruses jumping from species to species. This is not like first time we are observing it. This happening over the ages, and that is the natural phenomena, only because we have a better detection ability right now and we are able to detect them so effectively. But targeting a species just because it has been a source of certain infection, probably we need a very good awareness and more of a extension activities among the people who do so, rather than making bat itself as a culprit and deciding on vanishing them. So that probably needs some more of an awareness program.
Last
Okay, Girish sir.
question from Manasa. Okay, it's time up. Okay. So okay.
Sir, I will
We come to yeah.
I
Hello?
will take up quickly, sir. Okay. Girish sir.
Okay, from the YouTube side. Okay, please. Over to Abdul. Okay.
Okay, sir. Sir, coronavirus vaccine is available for animals.
For some of the coronaviruses, it is available, not in India.
It's through course of disease virus present in throat. Is throughout the course of disease virus present in throat.
During symptomatic and initial incubatory period, the virus is detected. Once the immune response is mounted, the virus is.
Number of passages taken to make it inactive.
That kind of studies is not done so far. Probably that will be a future observation which will get research progress.
Are people who recovered from SARS or MERS show some resistance to this virus?
Obviously, there will be a kind of cross protection to some extent, but they are not totally immune to the infections.
How long recovered person can maintain his or her immunity?
So far, the studies have shown up to 2 months, 3 months period during which the infection did not occur. But again, there are repeated infections which have been observed. So it is not long-lasting immunity.
What is the effectiveness of thermal screen?
Thermal screen effectiveness is not very good in terms of thermometer, but that is the only effective tool what we have now.
Okay, sir. Do you recommend reusable or washable mask for regular work? If yes, how long?
No, sir. Especially surgical mask, identify no, but the cloth mask which we are doing, those can be washed.
Any post-viral infection of corona?
Post-viral infection. So that basically I feel like reinfection. Reinfection is possible. It has been reported.
What is genetic similarity of SARS-CoV-2 strains circulating in India with one that was associated with index case?
So that information is very similar actually, but I don't remember the number as such, but I can share you.
What is the treatment regime of China is following since the cases are quite low?
China, the cases are low because of other intervention strategies, but not the treatment. The treatment is mostly symptomatic based on the symptoms they have been treating it.
What can be the sequel of disease?
So far, there are a number of reports to show this one, but none of the sequels are serious enough to be considered.
Something about Bharat. Okay. Something about Bharat Pharmaceuticals vaccine.
Bharat pharmaceutical vaccine. No, I am not aware of much.
What is the future of corona in India? This is the last question, sir, probably.
Future of corona, it will increase. The number of cases what we are observing is still very minuscule, and we haven't reached the peak, and it will increase. Hopefully, because of the kind of population we have, the mortality rate will be very low.
Okay, sir. Okay, Giri sir, please take over.
Giri sir, for concluding remarks. I think he lost the activity.
Sir, Giri sir is not present. Please continue.
Yeah, let me. So it is a privilege to have with us Dr. Jagadish Hiremath, who is the eminent scientist at ICSR Nivedi, who has worked in this area. He is working on the porcine, other kinds of porcine, and he is also in coronavirus. I think we are the right person to deliver right talk at the right moment. So today is World Zoonoses Day. So thank you very much, Jagadish Hiremath, for your. He of course Alumni of University for your valuable time on behalf of our university. The appliers, I extend heartfelt thanks to Dr. Jagadish Hiremath, sir. Thank you.
Thank you, sir. Thank you so much.
We are immensely. Secondly, we are very much immensely. We have our gratitude to express to our honorable Vice Chancellor for his valuable time that he spent in supporting our program, and in spite of which he sent his message that has been read by our moderator. Thank you, sir. And any deans, directors, and university officers and faculty members of our university and other university as well. So we thank all of them.